Hey guys, in this video we're going to learn how messenger RNA is modified post-transcription. So I'm sharing my screen with you. These slides are in chapter 10 on Blackboard. So we're going to talk about what happens to the messenger RNA, how it's modified after we make it, and then we're going to define these two terms, introns and exons. So let's look at how this messenger RNA is changed after we make the messenger RNA copy from our DNA. So here's our start of our process. We had the DNA in the nucleus. Remember, there was a special region called the promoter. That's where the RNA polymerase knew where to bind, where the start of the gene was. The RNA polymerase went along reading the DNA bases, making a single-stranded messenger RNA copy. And then when it got to the terminator region, it knew that that was the end of the gene, and then it stopped making messenger RNA at that point. So after the process of transcription, we have made a messenger RNA copy of our DNA. But the problem is there is actually too much information in this copy. So we're going to see that this piece of DNA that we copied is made of exons and introns. Exons and introns. Now the key thing is you only need the exons to make your protein. The intron is like filler that's in between. We don't need this to make our protein. So when we want to make our protein and finish making our messenger RNA, we don't need this piece, the intron. It's called an intervening sequence. We don't need the intron, so we're going to splice that out, and then we're going to be left with our exons, our express sequences stuck together. So that's one thing we have to do. We have to get rid of this extra data, these introns. We don't need them to build our protein. So we have to splice those out, chop them out, get rid of them, and at the end we're going to be left with a chain of expressed sequences. They're the recipe for how to make our protein. Now, the second thing is the express sequences have really valuable information. So when this piece of messenger RNA goes out to the cytoplasm, there are many enzymes out there that could digest and damage the messenger RNA. So we don't want a protein that's missing some amino acids. So we're going to add a cap and a tail to our messenger RNA. And basically the job of the cap and the tail is to protect the ends of the messenger RNA. We're adding some extra bases at the start. We're adding some extra bases at the end. These are going to protect our messenger RNA and make sure it doesn't get digested by enzymes that are out in the cytoplasm. You'll notice that the tail is very often a chain of adenines, A here. A lot of the time the tail is made of adenines, so it's called a poly-A tail. So many A bases all in a row, that's usually what makes up the tail. So we're changing our messenger RNA, we're modifying the ends to protect it, we're adding the cap on the 5' end, we're adding the tail on the 3' end, and the function of both of these is the same. They're going to protect our messenger RNA and stop it from getting broken down by enzymes. So then, like I said, when you copy your DNA and you make your messenger RNA, you get pieces you need, which are called exons, and then you get pieces you don't need, which are called introns. So you want to splice out the introns, get rid of these sections that you don't need, so your final messenger RNA should only contain your exons, the expressed sequences. So the intervening sequences, the junk that we don't need, those are called introns. The parts of the gene that we do need to make our protein, to express our protein, are called exons. So the exons are the important parts. The introns we don't need to make our protein. So the introns are removed, and then your final messenger RNA that's going out to your ribosome, that's going to be all exons, all express sequences of DNA. So here you can see the whole process, a nice picture from a textbook. You start off with DNA. Remember, there's going to be a promoter and a terminator region telling the RNA polymerase where the gene starts and where the gene ends. You're going to make a messenger RNA copy of your DNA. So you're going to faithfully copy every part of this DNA initially. You're going to copy the introns and the exons. So you make an exact copy of your DNA. You add your cap to protect the 5' end. You add your tail to protect the 3' end. Then you have to get rid of the introns. Remember, these are the areas that we don't need. So we splice them out, and splice is just a fancy biology word for chopping. So we chop up our messenger RNA and we splice out or chop out the introns, the intervening sequences, the parts that we don't need. So the introns get spliced out, and then at the end, we're left with our mature RNA, which has a cap, which has a tail, and which is all exons. So this is going to be going out of the nucleus to find the ribosome. So after it's transcribed, messenger RNA is modified. So this is called the post-transcriptional modification of messenger RNA. Okay, so hopefully you understand why we needed to do those steps. And then when we get our next video, we're going to learn how to do translation, how to make a protein from these recipes that we've just